Hi all, welcome to Physio Exam Buddy. This video is on regulation of the blood pressure and that too short-term regulation of blood pressure. By watching this video, you will be able to answer some of the previously asked university exam questions like neural regulation of blood pressure, short-term regulation, role of baroreceptors in blood pressure regulation, Bainbridge reflex, Basal-Jarisch reflex, as well as Cushing reflex. The first three can be asked as a short essay question, whereas the last three, that is 4, 5, and 6th question can be asked as an answer briefly or physiological basis questions. We will start about the regulation of the blood pressure. Arterial BP, it is regulated by several mechanisms which will try to maintain the mean arterial pressure within a very narrow range of 90 to 100 millimeters of mercury under physiological conditions. So, the mechanisms controlling arterial pressure can be grouped into three, that is short-term or rapid mechanism, intermediate term mechanism, and long-term mechanism. This video is on short-term or rapid mechanism. Short-term regulation of blood pressure. It is also known as neural regulation of blood pressure, as all the mechanisms are nervous type of reflexes. These mechanisms are also known as short term because they act very rapidly, that is act within seconds to few minutes of alterations in the blood pressure. And it also got a short duration of the action. So these three are the peculiarities of the short term or neural regulation of blood pressure. So these are the mechanisms coming under short term. First one is you have to explain the baroreceptor reflex, which operates when the mean arterial pressure varies between 60 to 180 millimeters of mercury. Second mechanism is the chemoreceptor reflex, which operates when the mean arterial blood pressure goes below 80 millimeters of mercury. And CNS ischemic response operates when the mean arterial pressure goes below 60 millimeters of mercury. And the most powerful and the potent mechanism is the CNS ischemic response, and it acts very powerfully when the mean arterial pressure lies between the range of 50 to 20. So that all these things will bring back up our mean arterial pressure back to the normal narrow range, that is 90 to 100 millimeters of mercury. So, in short, short-term regulation means it acts very rapidly, that is within seconds to few minutes of alterations of blood pressure. It also got a short duration of action, and it mainly includes nervous reflexes, that is baroreceptor reflex, chemoreceptor reflex, and CNS ischemic response. So, it is also known as neural regulation of blood pressure. So, if the question is neural regulation of blood pressure, there is no need for getting confused. It is just our short-term regulation of the blood pressure. Before going to detail about the nervous reflexes and the mechanisms of this short-term regulation, we should know what is the medullary cardiovascular center or vasomotor center. These are the two diagrams you are supposed to draw along with the baroreceptor reflex in short-term regulation. Both these diagrams are taken from the Gano. So there are certain parts which are important in this medullary cardiovascular center or vasomotor center. Four parts is important. First one, that is the nucleus tractus solitarius, which is the sensory area. Second part is the RVLM or the rostral ventrolateral medulla. And the third part is the CVLM and IVLM, that is the caudal and intermediate ventrolateral medulla. And the fourth one, it is shown in this cut section, that is cardio-inhibitory center, which include two parts. First one is the dorsal motor nucleus, and the second portion, that is nucleus ambiguous. So the four portions which helps in the control of the arterial blood pressure, which is situated in the medullary cardiovascular center or vasomotor center, is first one, that is rostral ventrolateral medulla or RVLM, then caudal and intermediate ventrolateral medulla, that is CVLM and IVLM, nucleus tractus solitarius or the sensory area, that is the NTS, and the cardio inhibitory center, which is divided into two, that is dorsal motor nucleus as well as the nucleus ambiguous. So this is the vasomotor center or the medullary cardiovascular center, and we have seen that RVLM or the rostral ventrolateral medulla. You can see here that the RVLM, it is located bilaterally in the upper medulla in the anterolateral region, and it mainly contains glutaminergic neurons. Means the neurotransmitter secreted from our RVLM is glutamate. Glutamate, it is an excitatory type of neurotransmitter, and one peculiarity of this RVLM is that it is like our pacemaker of the heart, that is, it discharges rhythmically in a tonic fashion, and it will excite the sympathetic preganglionic neurons in the intermediolateral gray column of the spinal cord. So RVLM secrete glutamate, which will excite the sympathetic preganglionic neurons in the intermediolateral gray column of the spinal cord, and from here the postganglionic neurons supply the heart as well as blood vessels. So the impulses from this RVLM, it is actually stimulating the sympathetic preganglionic neurons. So it causes sympathetic stimulation of the heart as well as the blood vessels, and these impulses maintain a partial state of contraction of the blood vessels, which is known as the vasomotor tone or the vasoconstrictor tone. Next portion of the vasomotor center, that is the CVLM IVLM. It is again located bilaterally in the lower half of the medulla in the anterolateral region. And the peculiarity of the neurons in the CVLM and IVLM is that it secretes the neurotransmitter, that is the GABA. And GABA means it is an inhibitory neurotransmitter. So GABA coming from the CVLM and IVLM will inhibit the RVLM. So it will cause a decrease in the sympathetic discharge to the heart as well as blood vessels. That is, it causes vasodilatation. Next area is the nucleus tractus solitarius or the sensory area. Location, you can see here, it is bilaterally located in the posterolateral portions of the medulla as well as in the lower pons. And this area, it also receives afferents from the glossopharyngeal or the 9th cranial nerve and the vagus or the 10th cranial nerve. And this will again send the impulses to the CVLM, IVLM and also into our cardio-inhibitory center. So NTS, the neurotransmitter secreted is glutamate, and it will excite the CVLM, IVLM and also cardio-inhibitory center. So whenever the nucleus tractus solitarius is stimulated, this glutamate causes activation of CVLM and IVLM, which causes secretion of GABA, which will inhibit the RVLM and decrease the sympathetic discharge and causes vasodilatation. Along with that, it also causes activation of the cardio-inhibitory center. So vagal stimulation will get activated. And these medullary cardiovascular center, it is regulated by the higher centers, that is hypothalamus, limbic system, reticular formation as well as cerebral cortex. Now we will see in detail the mechanisms of the short-term regulation. First, about the baroreceptor reflex, which operates in a range when the mean arterial pressure varies between 60 to 180 millimeters of mercury. The other name for baroreceptor reflex is sino-aortic mechanism. So question can also be asked as explain sino-aortic mechanism. This is mainly mediated by the baroreceptors. Baroreceptors means they are stretch receptors which are located in the walls of the heart as well as in the adventitia of the blood vessels. And baroreceptors means they are stimulated by distension. So whenever there is an increase in the pressure, it causes distension of the baroreceptors. That is, there will be increased stretch in the baroreceptor, which will increase the rate of discharge from these baroreceptors. And there are two groups of baroreceptors in our body, that is high pressure receptors as well as low pressure receptors. High pressure receptors monitor the arterial circulation and they include carotid sinus, aortic arch and walls of the left ventricle, whereas the low pressure receptors, they are located in the cardiopulmonary area. That is why it is known as cardiopulmonary receptors, and they include receptors which are located in the walls of the right atrium, at the entrance of the superior vena cava as well as inferior vena cava, and also at the junction of the pulmonary veins and left atrium and in the pulmonary circulation. First, we will see in detail about the carotid sinus as well as aortic arch. Regarding the location, Carotid sinus, you can see here that this is the common carotid artery which bifurcate into external as well as internal carotid artery, and the internal carotid artery just before the bifurcation, it got a small dilatation, and this is where our carotid sinus locates. Whereas aortic arch, which is located in the adventitia of the arch of aorta. Regarding the innervation of the carotid sinus as well as aortic arch, carotid sinus, it is innervated by a carotid sinus nerve, a distinct branch of the glossopharyngeal nerve, and the aortic arch, it is supplied by the vagus nerve. And these nerves, that is glossopharyngeal as well as the vagus nerve, they are commonly known as buffer nerves, as these are involved in buffering the blood pressure, that is preventing the sudden fall and rise in the blood pressure. Whenever a person is having normal blood pressure, these buffer nerves discharge only at a very low rate. But whenever the blood pressure increases, there will be increase in the rate of discharge through the buffer nerves. And whenever there is a decrease in blood pressure, which will also decrease the rate of discharge through these nerves. So whenever the baroreceptors are activated, it mainly responds to the BP variation where the mean arterial pressure ranges between 60 to 180 millimeters of mercury. And even a slight change in the blood pressure will produce the strong change in the baroreceptor reflex. And it mainly responds more to the rapidly changing pressure. So whenever there is an increase in the BP, it will cause increase in the distension of the baroreceptors or produces increase in the stretch in the baroreceptor, that is the carotid sinus and the aortic arch. So there will be increase in the discharge rate through our 9th as well as the 10th cranial nerves, which will secrete the glutamate. Glutamate, again, it is an excitatory neurotransmitter which will cause a stimulation of the nucleus tractus solitarius. So when the nucleus tractus solitarius stimulates, it also secretes the neurotransmitter GABA from the NTS, because there are glutaminergic projections from the NTS, and it will stimulate the CBLM and the IVLM. And whenever the CBLM and IVLM is stimulated, it will secrete GABA, which is a neurotransmitter from the CBLM and IVLM, and it will inhibit the RVLM, and this will decrease the sympathetic discharge towards the heart as well as the blood vessels. So normally the RVLM is secreting glutamate neurotransmitter, which will cause a sympathetic stimulation of the heart as well as the blood vessels. But whenever this RVLM is inhibited by CVLM and IVLM, it will decrease the sympathetic discharge to the heart as well as the blood vessels and produces vasodilatation and decrease in the blood pressure. And excitatory projections from the nucleus tractus solitarius, that is whenever the 9th and 10th cranial nerves stimulate our nucleus tractus solitarius, the excitatory projections also is sent to the cardio inhibitory center, that is the dorsal motor nucleus of the vagus as well as the nucleus ambiguus. So the excitation of the NTS is transmitted to the cardio inhibitory center, which will excite the vagal innervation to the heart, which will causes vasodilatation as well as venodilatation, and again decreases the blood pressure. And whenever the decrease in arterial blood pressure happens, the exact opposite happens. That is, it will decrease the stretch of the baroreceptor, which will decrease the inhibition in our RVLM. So there will be an increase in the sympathetic vasoconstrictor discharge as well as decrease in the stimulation of the cardio inhibitory center. So there will be increase in the blood pressure as well as increase in the heart rate. So we have finished with the first two high pressure baroreceptors, that is carotid sinus as well as aortic arch. Next is the left ventricular receptors, and they are located in the endocardial surface of the left ventricle, again stimulated by ventricular distension, and the response is vagal bradycardia as well as hypotension. One effect of the left ventricular reflex receptors is the coronary chemoreflex. This is also called as Basalt-Jarisch reflex, and it can be asked as a one-mark question. So this is an experimental finding, that is injection of drugs like serotonin, veratridine into the coronary supplying the left ventricle will produce apnea followed by rapid breathing, hypotension and bradycardia, and their receptors are is actually found to be C fiber endings, and the afferents go through the vehicle and produces hypotension. This is also we can see in myocardial ischemia like conditions. So this Basalt-Jarisch reflex can also be asked as a one-mark question. We have finished with the high pressure baroreceptors. Now next is the low pressure baroreceptors. These low pressure baroreceptors minimize the BP changes in response mainly to the changes in the blood volume. So whenever the BP change is occurring because of the blood volume, this low pressure receptors will respond. So first about the atrial stretch receptor. They are located in the walls of the atria, and there are two types of atrial stretch receptors, type A and B. Type A atrial stretch receptor will discharge during atrial systole, whereas type B discharge late in the diastole, that is during the peak atrial filling. So whenever there is an increase in the distension of this atrial stretch receptors, there will be increase in the discharge from the receptors, which will produces vasodilatation and decrease in the blood pressure. So the atrial reflex control of the blood volume, it occurs by different mechanisms that we will see. That is, whenever there is a volume overload, there will be stretch of the atria, which will activate the atrial stretch receptors, and they will either increase the secretion of atrial natriuretic peptide, which will cause natriuresis and decrease in and loss of fluid through the urine, or it can also decrease the secretion of the ADH from the hypothalamus and decreases the reabsorption of water from the renal tubules. And another action of the increased stretch in the atria and increased discharge rate from the atrial stretch receptor is that it causes reflex dilatation of the afferent arterioles in the kidneys, which will increase the glomerulocapillary pressure, and it will increase the filtration in the renal tubule. So all these conditions will increase the fluid loss from the kidneys and bring back the blood volume back towards the normal, thereby bringing back the blood pressure back towards the normal. So it is also known as the indirect pressure controllers, because it is the blood volume which is the culprit which causes increase or decrease in the blood pressure. So by bringing back the blood volume back to the normal, it will bring a control over the arterial blood pressure. So these atrial stretch receptors, they are also known as indirect pressure controllers. And another action of the decrease in the ECF volume is that it will causes a decrease in the central venous pressure, which will decrease the firing or discharge from the atrial stretch receptors, which can actually increase the ADH or vasopressin and increase the aldosterone secretion. So whenever there is an increase in the vasopressin as well as aldosterone, it causes sodium as well as water retention and bringing back the normal ECF volume. And another action of the decreased ECF volume is that it can directly increase the sympathetic activity and increases the secretion of the renin and increases in the aldosterone secretion, which will again causes the increase in the sodium and water retention, bringing back the ECF volume back towards the normal. Next is about the Bainbridge reflex or the atrial reflex control of the heart rate. So whenever we rapidly infuse either blood or saline, there will be an increase in the heart rate if the initial heart rate of that individual is low. And the action is because of the atrial stretch receptor activation, which will stimulate the vagus. That is, whenever there is rapid infusion of blood or saline, it will cause a stretch in the atria. So the atrial stretch receptors will get stimulated, which will send the impulses via vagus into the medulla and increases the heart rate as well as force of contraction. And this mechanism, that is the Bainbridge reflex, is the one which prevents the damming of the blood in the veins, atria as well as in the pulmonary circulation. So we have finished with the baroreceptor reflex. Next is the chemoreceptor reflex, which operates when the mean arterial pressure goes below 80 millimeters of mercury. Chemoreceptor reflex. What do you mean by this chemoreceptors is that these are receptors which senses the decrease in PO2 or hypoxia, increase in PCO2 or hypercapnia, and decrease in pH, that is acidosis in the blood. So whenever there is hypoxia, hypercapnia and acidosis in the blood, it will be detected by the chemoreceptors. Then what are the chemoreceptors in our body? There are two types of chemoreceptors. One is carotid body and the second one is aortic body. Carotid body, you can see here that it is located near the carotid bifurcation and it got the innervation from the glossopharyngeal nerve and a branch of glossopharyngeal nerve which is known as the Herring's nerve. And the aortic body, regarding the location, it is situated near the arch of the aorta supplied by the vagus nerve. And these chemoreceptors act when the BP falls below 80 millimeters of mercury. So when the mean arterial pressure goes below 80 millimeters of mercury, commonly occurs in conditions like hemorrhage, there will be decrease in the blood flow to the chemoreceptors. So whenever there is decrease in blood flow, there will be decrease in the oxygen content. So hypoxia will be there, increase in the carbon dioxide content is there, that is hypercapnia will be there, and the hypercapnia will cause acidosis. So all these things will stimulate the carotid and aortic bodies and it will excite our medullary cardiovascular center or the vasomotor center and elevate the BP back towards the normal. So whenever the BP is elevated, it will improve the blood flow to the receptor organs and it will eliminate the stimulus of the chemoreceptors. And again, the BP falls and the new cycle is initiated. This is how the chemoreceptor reflex bring back our BP back towards the normal. So we have finished baroreceptor reflex, chemoreceptor reflex. Next is the CNS ischemic response, which operates when the BP, mean arterial pressure, goes below 60 millimeters of mercury. CNS ischemic response. It is an emergency pressure control system because they act very rapidly and powerfully to prevent the decrease in the blood pressure, and it acts when the mean arterial pressure goes below 60 millimeters of mercury, and the greatest degree of stimulation by the CNS ischemic response occurs at a pressure, that is mean arterial pressure, between the range of 15 to 20 millimeters of mercury. When the mean arterial pressure goes below 60 millimeters of mercury, there will be a decrease in the blood flow to the brain. That is, it will decrease as the cerebral blood flow, which will cause a buildup of carbon dioxide as well as lactate in the brain, which will activate our vasomotor center, presumably the RVLM, and stimulate the sympathetic activation and causes vasoconstriction and increase in the blood pressure. The reflex related to the CNS ischemic response is the Cushing reflex. That is, whenever there occurs a raised intracranial tension, it causes bradycardia. So, how the raised intracranial tension causes bradycardia is the Cushing reflex. Whenever there is an increase in the intracranial pressure, it causes compression in the arteries in the brain, which will decrease the blood supply to the vasomotor center, which will decrease the oxygen as well as increase buildup of the carbon dioxide. So this hypoxia as well as hypercapnia will stimulate the vasomotor center and causes an increase in the blood pressure, which will stimulate the baroreceptors and causes decrease in the heart rate by decreasing the sympathetic stimulation. So that is how the increased intracranial pressure will cause a decrease in the heart rate. That is the Cushing reflex. That is, any condition causing increase in intracranial pressure will decreases the heart rate, and this is the mechanism of the Cushing reflex. So to summarize, short-term regulation of blood pressure means short duration of action, act very rapidly, and it includes nervous reflexes. That is why it is also known as neural regulation of blood pressure, and it includes baroreceptor reflex, chemoreceptor reflex, and CNS ischemic response. Baroreceptor reflex operating in a range when the mean arterial pressure varies between 60 to 180, and you have to explain both the high as well as low pressure baroreceptors. And in chemoreceptor reflex, it operates when the mean arterial pressure goes below 80. And you have to explain the mechanism of the carotid and aortic body activation. Next is the CNS ischemic response. When the mean arterial pressure goes below 60 millimeter, please write that it is most powerful when the mean arterial pressure goes between 15 to 20 millimeters of mercury, and also explain the Cushing's reflex. So the role of baroreceptors in BP regulation, you have to explain both the high as well as low pressure baroreceptors. Only the baroreceptor reflex you have to be explained if the role of baroreceptor is asked as a question. Thank you for your patient listening, and keep on watching Physio Exam Buddy, which makes your physiology exams more easier.